Thank you. All right, so I should say from the start that I'm one of two island biologists in the state of Louisiana, one in Baton Rouge and one in New Orleans. And so I think the pancreatic beta cell, which is the only cell that makes and secretes insulin, is incredibly interesting because that's the route to get both type 1 and the type 2 diabetes. So we're going to do an overview today on how do pancreatic islets function normally, and then how do they change function in autoimmune states, which are related to type 1, and as well as insulin resistance and obesity, which is obviously relative to type 2. If the slides work.
You might just have to click on the slide.
Let's see. Good idea. Good call. All right, so there's a lot of slides you can use to make the point that diabetes is a major public health problem. I personally like this one because I think the CDC does a great job of pointing out that whether you talk about it in terms of millions of people with diabetes or whether you talk about it in terms of percentage of the population, the graphs look the same. In other words, it's going in a direction that people find alarming and is obviously a major public health problem. So much so that popular periodicals have picked up on this and are talking about the growing diabetes epidemic, diabetes are at risk, a growing health crisis, et cetera, et cetera. And they often just sort of conflate both forms of the disease and don't really delineate type 1 and type 2. But I think we can all agree that diabetes exists in two major forms, right? Type 1 is autoimmune, type 2 is closely related to obesity, and then there's a scattering, a small percentage that are monogenic forms. In other words, you have an inherited mutation that essentially causes a decrease in insulin secretion. Almost all of the monogenic defects and forms of diabetes occur in the pancreatic beta cell. And so your link between type 1 and type 2 diabetes, or your ability to get diabetes of any kind, depends on death and dysfunction of pancreatic beta cells. All right, so for anyone that's not familiar with this or needs a review from past courses, pancreatic islets of Langerhans are small, specialized endocrine structures housed within the bigger organ of the pancreas in general. Right? So the pancreas essentially has two functions. It has production of digestive enzymes, which are released into ductal systems for breaking down food molecules. That's about 98% of what the pancreatic tissue does. The other 1 to 2% are these specialized structures, in this case just showing one. This is one islet situated in a housing of what's called acinar tissue, or essentially non-endocrine tissue. This is human. This is what it looks like in the mouse. This is a picture taken from my lab. What you can see is acinar tissue out here and standing for insulin in red. Because whether it's a mouse, a rat, or a human, the predominant cell type in the pancreatic islet is a beta cell, which makes and produces insulin. Now there are other cell types. Alpha cells make glucagon, delta cells make somatostatin, and so on and so forth. But it's the hormone insulin that, if it's not present, you have hyperglycemia and the other clinical symptoms that are associated with diabetes. Okay, so what is the importance of this hormone? I think this is the one slide summary of glucose homeostasis and how pancreatic hormones are important. So in the fasting state, you would have glucagon from alpha cells secreted at a higher level. That's important for promoting glucose production from liver to sustain blood glucose at a normal level, 70 to 100 mg per deciliter in a healthy human. It also promotes fat breakdown from adipose tissue and release in circulation of non-esterified or free fatty acids. But after a meal, so you ingest some food material that's probably going to be a composition of protein, carbohydrate, and fat, so blood glucose levels would rise transiently. The beta cells sense this through a metabolic process that requires glycolysis, TCA cycle, metabolic molecules that promote an action potential, which I'll show you. And insulin secreted from the beta cells is required to shut off that hepatic glucose production and also accelerates glucose uptake in the muscle and fat. So it's this coordinated switching of metabolism that prevents the chronic rise in glucose that we associate with metabolic disease. Now the importance of the hormone insulin, I think, is showcased quite nicely here. This is some pretty old work 17 years ago or so in rat adipocytes, and shows that lipolysis is occurring at quite a high rate. So you got fatty acid breakdown, in other words, this would represent fasting state. And as you introduce a very small amount of insulin, you can rapidly shut off fatty acid release. And at the same time, glucose incorporation in the lipid is occurring. So this is showing that glucose is being taken up by the GLUT4 transporter, converted through glycolysis and other processes into lipid molecules, which are then stored. And so this reciprocal regulation of shutting off lipolysis and stimulating glucose uptake is incredibly important and does not occur in diabetes. All right, so let's look at how the beta cell secretes insulin in response to glucose. And the simplest way to think about this is it's analogous to a neuronal action potential, except instead of neurotransmitters, beta cells respond to nutritional stimuli with electrical activity, which promotes the exocytosis of the packaged insulin granule. So if you look at what we call the basal state, in other words, that's when blood glucose is less than 5.5 millimolar, or roughly around the fasting state, about 70 to 90 mg per deciliter. Glucose is still in the blood, so it's taken up, but it's not really stimulating the high Km hexokinase called glucokinase that's present in beta cell and liver. And so you don't get much trafficking or exocytosis of the granule. But when you ingest a meal or a sugar-sweetened beverage or any type of food source that contains glucose, the glucose value in the blood rises, there's more molecules that are then taken up, and that metabolic activity is coupled to ATP production, which closes an ATP-gated potassium channel. And this membrane depolarization essentially fires as an action potential, measured the same way you can measure an action potential in a neuron, and opens these voltage-gated calcium channels. In addition, these metabolic signals promote release of intracellular calcium stores. So the endoplasmic reticulum inside a cell concentrates calcium, and it can be released under certain conditions. In this case, in the beta cell, metabolic activity stimulates calcium activity in response to electrical activity. Together, these calcium signals promote vesicle trafficking up to the cell surface where they fuse with the membrane, and then they're released into the circulation. And this process is incredibly rapid. From the time of glucose being introduced to what's called first phase insulin secretion, it's about 2 to 5 minutes. So it's a really fast response. And this is what it looks like as a whole, just to remind people that this is a coordinated process, right? As glucose spikes, so this would be your fasting period, such as overnight during sleep, wake up and have some type of food for breakfast. Glucose rises transiently as long as you have insulin, and then is brought back down as production from liver is decreased, and transport and uptake is promoted in muscle and fat. And of course, there's also storage in liver as glycogen and lipid. You can see the opposite pattern happens for glucagon. It starts off high, whereas insulin is low in the fasted state, and as insulin goes up, glucose drops precipitously. This relationship is incredibly important for glucose to be maintained at a normal level. What happens, obviously, in diabetes is you don't have this signal, and so this signal stays high. In response to a meal, for example, glucose, glucagon can rise even higher. And since glucagon is capable of stimulating glucose production in the liver, what you get is an exacerbated situation where the glucose that's taken in through diet is not transported properly to tissues, and liver is still making glucose at a flat-out rate, right? And so you look at untreated diabetics, and blood glucose is 500 to 800 mg per deciliter, whereas it should be somewhere in the 70 to 100 range. All right, so if anybody is a member of the American Diabetes Association, you're probably sent every year the clinical practice guidelines. They're rarely updated, but in some cases they are. And this just gives you an idea of what the criteria for diagnosis of diabetes are. And you can see there's multiple. And there's no one preferred test. Right? So of the 1, 2, 3, 4 you see here, all of these can be used. In many cases, people use 2. For example, hemoglobin A1C greater than 6.5%, fasting plasma glucose greater than 126 mg per deciliter. You can use the oral glucose tolerance test, et cetera, et cetera. And the indication is testing after age 45 or earlier if there's a family history of diabetes. first degree relative to diabetes. All right, so why is the A1C used as a marker? And I think this is a pretty good illustration of why that would be. In this case, it's just showing prevalence of retinopathy, so diseases of the eye. Obviously, if you have chronic hyperglycemia, you're going to get predisposed to diseases of the eyes, kidneys, and cardiovascular system. And this is showing that as A1C levels creep up by percentage, the percentage of people with retinopathy gets incredibly elevated, and you see increased risk for kidney diseases and increased risk for cardiovascular diseases. All right, so that was the brief overview of how things work normally and why it's important for insulin to be secreted in a regulated fashion, how hormones from the pancreatic islets regulate overall blood glucose and metabolic homeostasis. But I think the goal here was really to talk about how things break down and how does this process get interrupted from two major forms of diabetes that start off with very different origins, right? Autoimmunity, which we'll talk about now, and obesity, which we'll talk about later. They get to the same endpoint, right? Death and dysfunction are the only cell type that makes and secretes insulin. All right, so there's a couple of major questions in the field. And when I say major questions, these are things that have been thought about for 50 to 100 years and not answered. One is, what's the initial trigger that leads to the immune system seeking out insulin-positive cells and getting rid of them? So your immune system normally does a great job, hopefully, of if you get a bacterial infection or get a viral infection, it goes through the innate immune and adaptive immune processes to eliminate those pathogens. That's good. That's the function you want. When it goes through the same types of processes and eliminates a self-tissue, that's not good. And that's when you have an autoimmune disease, of which type 1 diabetes is classified. Now, the initial trigger that leads to this has been looked at since people realized this was an autoimmune disease and never found, suggesting, in my personal opinion, that there's not a single trigger. There are probably more than one. The other question is, what controls immune cell infiltration into the pancreatic islets? Pancreatic islets, like many tissues, have resident immune cells. They're supposed to be there. They're important for the normal growth, function, and health of that cell. But when you start piling in 2 to 3 to 4 to 5 times that number of immune cells, and they're doing very different things, that's what you don't want. And so obviously in type 1 diabetes, there's an immune cell elimination of that self-tissue, so there has to be a reason, a biochemical logic, for why immune cells entered that tissue in the first place. And that's what my lab has worked on for the past 7 years. So to give you an idea of what this looks like, this is human tissue taken from cadaveric donors. And in this case, you've got an individual with no history of disease, and so what you're seeing is one pancreatic islet. This is the acinar tissue, or cell types that make digestive enzymes and other things that are non-endocrine related. Insulin is stained for in the red color. The glucagon-positive alpha cell is in maroon. And what you can see in someone that has had type 1 diabetes is a selective elimination of that insulin-positive cell. The other cell types in the islet are unaffected. The glucagon-positive cell is not destroyed. The somatostatin-producing cell is not destroyed. Only the insulin-producing beta cell. And so the development of type 1 diabetes can really be summed up in this type of schematic. You inherit some genetic predisposition, but this doesn't explain everything, because identical twins only get diabetes at about a 40 or 50% concordance rate, so it's not 100% genetic. There are putative environmental factors that people don't necessarily agree on, but it's accepted that a combination of environmental factors and genetic predisposition set up a situation where immune cells enter the islet, which is collectively referred to as insulitis by the immunologists and the islet biologists that study this. There'll be circulating antibodies that target typically beta cell proteins. And this immune dysregulation ultimately leads to a loss of that cell that produces insulin. And the individuals proceed through normal glucose tolerance, a period of glucose intolerance when they are starting to lose the ability to secrete insulin, and eventually full type 1 diabetes, which requires lifelong insulin administration for them to stay alive. All right, so to start with risk factor. Right now, at the present day, it's not understood what's the role of the autoantibody. Right, so B cells typically are involved in autoantibody production. but it's not clear if that's causal for the disease or just a biomarker for the disease. But regardless, it's an incredibly predictive factor for whether you're going to get the disease or not. This particular review came out last year, has plotted this as proportion of patients without type 1 diabetes. And so what you're looking at is if you don't have any autoantibodies, you essentially have no risk of developing type 1 diabetes. You develop 1 and you get a mild risk, 2, 3, and they didn't show 4. 4 is essentially you got a 100% chance of getting the disease. All right, so we know some of the things that are critical for human onset of type 1 diabetes. And obviously to get down to basic biochemical and immunological mechanisms, you have to have model systems which you can carefully manipulate to figure out key components of this disease structure. I've listed 4 of these in broad categories. I put the NOD mouse as a start. It's what's called the non-obese diabetic mouse, so it stays lean, but it develops diabetes with the same type of spontaneous onset as you see in human, and with a lot of the same genetic risk factors, the same genetic loci that are important for humans to develop. This mouse model also has it. So that's the gold standard for drug companies. If they want to test a new drug, they'll go to the NOD mouse first. If it works there, they'll start trying to scale up to primate and then obviously to do a clinical trial. The biobreeding rat is essentially the rat version of this mouse. It's used less frequently, probably because it's about 10 times more expensive. The multiple low-dose streptozotocin injection model. Streptozotocin is a chemical that you can inject into mouse or rat or monkey and get an inflammation in the pancreatic islets, get the death and destruction, get the hyperglycemia, and essentially control the onset. Because in the first 2 models, you can't control the onset. So some mice are going to develop at 10 weeks of age, some mice are going to develop at 14, some at 20, some never. And so while that does mimic the heterogeneous nature of human disease, it can be problematic for certain research projects. This model allows you to time it anytime you want them to become diabetic. And then I believe Dr. Mine had already talked about transgenics in a broad sense, but certainly there's thousands of transgenic models with more to be developed. Manipulating one gene versus another, and that can be a knockout, that can be an overexpression, that can be crossing 2, so you have 2 genes knocked out. Obviously, as you start to drill down on genetic loci that are important for immunological complications and biochemical outcomes, you can make mouse models that target specific regions. All right, this gives a reasonable overview of the characteristics of type 1 diabetes across mouse, rat, dog, and human, because obviously a variety of preclinical and clinical studies warrants cross-comparison, because if you're studying a disease in a mouse model, you want to be sure that whatever model you picked most closely represents the human disease outcome. And so what you can see is pretty faithfully, genetic susceptibility is a component across the spectrum. Autoantibodies are a component across, more variable in the dog because there's various breeds, and so some develop more frequently and some develop less frequently. Encephalitis is a feature. Encephalitis obviously just means immune cell infiltration into the pancreatic tissue. Whether it's an inbred or outbred strain matters. Most mouse strains that people use universally are inbred strains, so that each mouse closely genetically resembles the next mouse in line, so you can get as homogeneous an outcome as possible, even though with any living organism you're going to have variability. The sexual dimorphism only occurs in the mouse model. In other words, in this case, the females develop it more frequently than the males. That's not true in the rat, dog, or human. Ketoacidosis, variable in the mouse, less variable as you get larger in species. Across the board, if you don't have insulin and you go untreated, your lifespan is going to significantly shorten. All right, so let me show you a little bit of what this looks like in the mouse, and then we'll compare it to the human. What I've done here is pick some images from my lab, and the BALB-C mouse and the C57 Black 6 mouse are what you consider standard mouse strains. They don't have any inherited diseases. They're not predisposed to diabetes. If you just leave them in a cage and let them live, they'll live a long time, and they usually won't develop diabetes. So what you see is in this mouse model, we're staining for CD3, which marks T cells. So it's part of the adaptive immune system. A normal islet does not have the presence of T cells. And that's true in the mouse, that's true in the rat, that's true in the human. The NOD mouse, which spontaneously develops type 1 diabetes, by 8 weeks of age, they've got what's termed periinsulitis because it's kind of polarized to one side. By 12 weeks, they have what's called invasive insulitis. In other words, the immune cell has infiltrated the islet. Right? As you can see in these models, it's not supposed to be there. At this time point, these mice are non-diabetic. If you just measure their blood glucose, or you take their body weight, or you measure they look just like these mice. You can't tell them apart, except when you start staining for immune cell infiltration. So they're developing a slow, silent disease process, which is exactly what appears to occur in humans. It's clinically silent until hyperglycemia ensues, as well as other symptoms. If you look in the human, these are 4 different cadaveric donors of disease duration, diagnosed 1 year of diabetes, 3 years after diagnosis, 7 years, 8 years. Same type of thing, except they've stained for insulin in green, so you can see there's still insulin positivity throughout these particular islets. I don't know if you can see it here, but there's red dots which represent T cells. Right? So same type of thing in human, you get T cell infiltration into pancreatic islets. Same thing in mouse, same thing in rat. In the normal, non-diabetic across species, you don't have T cell accumulation. All right, so one of the things that my lab and other labs worldwide have been working on is what would tell a T cell to leave the blood and enter the pancreatic islet. There's got to be some biochemical signal that tells them this is the place you need to go. And Peter Hitveld put out an interesting commentary in 2009, essentially saying, why would any immune cell, as it's going through the blood, stop and attach to the blood vessel wall and then squeeze through and enter in pancreatic tissue, and then not just enter into pancreatic tissue, right, because 98% of pancreatic tissue is non-endocrine, but find the endocrine cells. How does it know to go there? And the answer was unknown, even though people knew there is some conversation, if you will, between the immune system and pancreatic islets. So what my lab did in 2010 is asked a question, okay, what if we just screen the entire rat genome and the entire human genome under a situation where there's no inflammation, in a situation where there's inflammation? Because characteristic of type 1 diabetes is pancreatic inflammation. Select for highly expressed genes, and we particularly, out of those 30,000 genes, you have to have some way to cull the list down to something manageable. I was particularly interested in secreted factors. Why? Because the beta cell secretes something, and it goes into the blood. And the immune cell sees it and follows it up a concentration gradient. That would be one possible way the immune cell would know where to go, right? We had no idea if the beta cell made any secreted factors other than insulin, right? And were any of these factors important for immune cell recruitment? And then, as my background is in transcriptional regulation, we were interested in things that were highly expressed. What genomic response elements controlled that activity, and how could it be regulated up and down like a dimmer switch? Right? And this, in my opinion, would allow us to understand the coordinated changes that control beta cell immune cell interactions. All right, so what we found, and what I'm going to show you is just a list of, if you're not an immunologist like I wasn't, a series of random letters and numbers. And so when this came out of our screen, we were a little, I want to say disappointed, but overwhelmed because these were molecules we had no idea what they did. Our screen was rat. Human, Dirk Homan, who's an immunologist, did a similar study in mouse human, and this is the collated list between what we found and what he found. So what you're seeing is mouse, rat, and human, with the idea being if it's across species, it's most likely to be conserved, and thus an important biological mechanism. So what we found were a series of proteins. These are indeed small secreted proteins that control macrophage recruitment, that control neutrophil recruitment, that control T cell recruitment. And when we saw a group of proteins that controlled T cell recruitment, we knew we were really onto something. But the fact that almost every single immune cell has a signal that beta cells can make and recruit it into the islet was incredibly important for the immunological outcomes. So just by way of a brief introduction, if you've never heard of a chemokine before, the immunologists have decided that the way these should be named is the placement of an N-terminal cysteine amino acid residue. If there's one cysteine in the N-terminus, it's in the C family. If there's two side by side with no other amino acid between it, it's in the CC family. If there's any amino acid separating it, which they call here X, it's in the CXC family, and so on and so forth. There's about 50 of these molecules that hit about 20 different receptors. So a really broad family of proteins that signal via cell surface receptor. These two together make up the bulk of the family. So if we go back and look, what you can see is the CC family and the CXC family are indeed highly represented in mouse, rat, and human islets that have undergone some exposure to inflammation. What's also incredibly important is that if you cluster these to their mechanism of action, all three of these proteins signal through this receptor. This receptor happens to be clustered on neutrophils, which are an important innate immune cell. You don't need to worry about that necessarily. All three of these hit this receptor, which cluster on T cells. So what's the end result? Beta cells can make proteins that can essentially recruit any immune cell into the pancreatic islet. And this will give you a sort of a global idea that you get an interplay between almost every single type of immune cell in type 1 diabetes. And so you don't have to be an immunologist to appreciate the fact that this is a highly coordinated immunological process. Started off early by neutrophils, which beta cells can recruit. They secrete proteins that stimulate the plasma cytoid dendritic cell to secrete cytokines. Conventional dendritic cells essentially present antigen. They're telling T cells what to go look for. In this case, it would be a self-peptide. Many of these T cells in both mouse and human, if you pull them out of a type 1 diabetic patient, are primed and targeted against proinsulin. And then the macrophage, which is not shown on here, is typically present already in pancreatic tissue, but it is in a state that secretes a large number of toxic molecules, such as cytokines, that essentially kill this self-tissue. And so beta cells are essentially secreting high levels of molecules that recruit all of these immune cells in through an unknown trigger. I'll give you an idea of the importance of some of these receptors. So the CXCR1-2 receptor, if you use pharmacological inhibitors in a clinical trial, you can enhance pancreatic islet survival after transplantation, showing that beta cell production of these molecules that are allowing beta cells and immune cells to interact, and those immune cells reject that transplanted tissue. If you can block that interaction, you can prolong survival of that transplanted tissue. The crosstalk between neutrophils, this essentially was in Nature Medicine because it was the first study to show that innate immune cells really were part of the priming response that promoted the eventual T cell macrophage mediated killing. So all the action, if you will, had focused on the T cell response for the last 30 years. And this really brought to the forefront the role of the innate immune cell. And then really a kind of an observational study pointed out that if you look in the blood of new onset diabetes, what you saw is circulating neutrophils went down, but if you had access to that donated tissue, neutrophils within pancreatic tissue went up, right? So they're leaving the blood, they're entering the pancreatic tissue. That stimulated some interesting additional research where people figured out the same thing happens in the mouse, right? So this is a cross-species mechanism. All right, so if we go back to how do immune cells enter the pancreas, which has been an ongoing question in the field for the last 30 years, what signals leukocytes to leave the blood? Our lab and others have really pointed out that it's this production of these small secreted proteins called chemokines that are important for recruiting a variety of immune cells into that pancreatic tissue. And so these are presented in different colors because each different protein has a different immune cell it selectively recruits. Right? So the ones that are recruiting CXCR2 on neutrophils, T cells ignore that signal. And the chemokines that recruit T cells, neutrophils ignore that signal. So it's a highly coordinated and specific response that's just beginning to be understood. So this just shows you in some of the models that we talked about at the first, the injected streptozotocin model, all the chemokines that you see in the human tissue are elevated in that particular model, which people often use as a type 1 model. In our studies in NOD mice at 10 weeks, and we specifically use 10 weeks because they're non-diabetic, because if this response was to coordinate immune cell entry into pancreatic tissue that eventually led to diabetes, they would have to be elevated prior to disease onset for that to be true. And so what this is showing is that anywhere from 10 to 100 to 500-fold increases in a different protein that beta cells are making that recruit immune cells are indeed elevated prior to diabetes onset. And then the other reason we chose 10 weeks is if you take 500 NOD mice and you map their onset, almost none of them have diabetes at 10 weeks of age, but they're right on the fringe because they'll start to develop at low frequencies at 11 or 12 weeks and much higher frequencies above 20 weeks. And you probably can't see it, but the average is sitting right here. This is at the University of Florida, about 16 weeks of onset. We average about 18 weeks of onset, so pretty good agreement. All right, so just to give you a flavor of other labs working on chemokines, there's a variety of publications, not just in type 1 diabetes, but looking at islet transplantation, looking at type 2 diabetes, and looking at various both rodent models as well as actual human tissue. And then the last 8 or so years, there's been a number of publications linking proteins that control immune cell responses with pancreatic inflammation with disease onset. All right, so the last thing I really want to show, a couple of slides for type 1 diabetes, and we'll move on to the obesity component, is what happens to insulin secretion in pancreatic islets in people with type 1 diabetes. Right? So you have all this immune cell infiltration, which is not supposed to be there. In the normal, non-diabetic, healthy individual, that immune cell response and infiltration is not there. So if you look at the ability of their islets to secrete insulin, just remember that if you add glucose, you get insulin secretion through these processes. You can also add other signals that will depolarize the membrane. Potassium chloride or the amino acid arginine are often used. If you look at the responses of the pancreatic islet in healthy humans, which is shown in the dashed line, if you have low glucose, they're not secreting much insulin. If you raise it, they'll start to spike. If you raise it and add an additional stimulatory signal, in this case glucose plus arginine, it spikes higher. You can put it back down to normal, they'll go back. So as soon as they see high glucose, they secrete insulin. If you take glucose away, they stop. If you give them glucose again, they secrete. If you take it away, they stop. What you can see in the black line is the type 1 diabetic islet. It's largely unresponsive to most of the signals. And my lab has focused a lot of our attention on how do you go from this level of secretion to this level of secretion. And it turns out cytokines produced from immune cells will generate this response. Right? So the immune cells are coming in and releasing things that essentially impair insulin secretion. All right, so there's some unknown trigger that's been sought after for 80 years, still unknown. We and others have been able to point out that chemokine production from beta cells is important for recruiting all of these immune cells, and the arrows show that these immune cells also interact with each other within the pancreatic islet, with the end goal being a decrease in beta cell mass and function. And so if you can short circuit these responses, we think you can prevent getting from here to here. But part of that problem in the human situation is diabetes onset is when people start to get treated, right? You're not experiencing symptoms, then all of a sudden you are, you go in, you find out you have been diagnosed with diabetes. Here's our current target period for clinical trials, way down here. Beta cell function, 80 or 90% lost already. The idea would be you want to intervene earlier. If you can figure out what the inciting event is, which no one knows, or at least figure out biomarkers for what is going on, we think increase in chemokine production is probably going to be one, because this beta cell loss is silent. And so Dr. Daniel Shaw and I have been working on detecting cell-free DNA from beta cells early in adolescent populations to find out, can you detect here and potentially intervene here rather than here? Right now we don't know the answer, but it's at least one attempt to try and get at the question much earlier before disease is actually developed. All right, so I'll stop on the autoimmunity component and move on to the obesity component. And just to give, I think that most people in this room probably think much more about type 2 diabetes than type 1. I know the Pennington is really focused on obesity and insulin resistance, and type 2 diabetes really as an end goal. What I'll do is just say some things that I think are relevant, which are it's the most common form of the disease. Probably if you hear about diabetes, 80 to 90% of the cases you're going to hear about is going to be type 2. Obviously, this is closely mirrored by rise in obesity in the general population. But you can be obese and insulin resistant and never become diabetic. In fact, that's probably what 75% of people fall into, right? Insulin resistance and diabetes are not the same thing. Insulin resistance is its own condition, which you can persist in for years, decades. If you develop a second defect, and that's an inability to secrete adequate amounts of insulin, that's when you'll develop diabetes. All right, so this will give you an idea. This is from Ron Kahn at the Harvard Medical School, where he did, I think, a pretty reasonable summary of all the things people think contribute to type 2 diabetes and insulin resistance. And I think most of us probably really focus on the components associated with obesity, so obviously over-consuming calories, not moving around enough. If you start storing lipid and lean tissues, which is not good, but it's supposed to be stored in adipose tissue. If you start forcing lean tissue, such as liver or muscle or beta cell, to store fat and do its normal function, it's not going to be good at its normal function. This also creates an inflammatory reaction, which we know is associated with insulin resistance. However, this component here is not sufficient to lead to diabetes. You have to have a second defect, and that defect's inadequate insulin secretion, which is often coupled to beta cell death. That's how you get type 2 diabetes. And so to give you a contrast or a comparison, you saw the islet in a human in a type 1 case. This is the islet from a human in a type 2 case. So again, healthy individual, no history of disease, acinar tissue out here, insulin stained for in maroon, and what you can see is the normal complement of insulin-positive cells in the islet. In the type 2 diabetic islet, what you can see is it's larger, and you can tell the picture from here to here looks vastly different. The pancreatic tissue has a more fibrotic state in the obese condition. The islet typically expands during insulin resistant and is usually full of insulin-positive cells. But by the time someone has progressed all the way to type 2 diabetes, what you can see is they have much fewer insulin-positive cells. And the decrease in insulin-positive cell mass is replaced by essentially fibrotic tissue. All right, so this essentially just shows that when someone's lean and then they start to increase their caloric intake, at first that increase in carbon consumption is going to be stored in adipose tissue, right? So you get an expansion of adipose tissue mass, which most people don't like for cosmetic reasons. But for health reasons, that's where you want the fat to go. Once you've exceeded this capacity, and you have to start putting fat here and here and here, that's what you don't want. And that's when you start getting metabolic syndrome and then progressing to symptoms of type 2 diabetes. Almost every single tissue in a type 2 diabetic state is affected by inflammation in some capacity, right? Whether you forced it to store lipid or you forced it to deal with an altered metabolic situation that comes with overnutrition and sedentary behavior. Immune cells will show up, or the lipid itself will create a chronic low-grade inflammation, and that tissue will function differently than it does in a lean state, including pancreatic beta cells, which will become defective in insulin secretion. All right, so just like in type 1 diabetes, type 2 diabetes has many rodent models that are incredibly useful to understanding disease mechanisms relevant to the human condition. Many people use the Zucker diabetic fatty rat. These are a monogenic form, so they have deficient leptin signaling, they overconsume calories, they get incredibly fat, and they experience all progression through insulin resistance to type 2 diabetes. You can high-fat feed the mouse or the rat. You can use the DBTB mouse, which is for all intents and purposes the mouse version of the Zucker. And just to give you an idea, these are litter mates, right? So siblings. If you have one copy of the mutation, you're still lean. If you have two copies of this mutation, the mouse is incredibly fat. And you can see this clever picture that was published probably 20 years ago now. You got one litter mate on the scale and two leans, and he's weighing down, he's twice the weight, more than twice the weight of those combined, which is exactly what we see when we use these mice. So there's a variety of basically fat mice and fat rats that are individually interesting to get at different questions through insulin resistance and spectrum that lead to type 2 diabetes. And just to give you an idea of, if you start looking at metabolic phenotype in the obese rodent and compare it to what we know about metabolic syndrome in humans. Well, obviously they're both obese. That's key. You get elevated blood lipids in both. In the rodent, it's typically referred to as atheromatosis disease, but oftentimes you see clinically it's called coronary artery disease in the human. They're essentially the same thing. They both get fatty heart. Hypertension is a little bit more variable in the rodent, not so much in the human. They get insulin resistant. If you measure glucose tolerance, they both have glucose intolerance. And then obviously the ones that progress to full type 2 diabetes in both cases have defective pancreatic beta cell mass and function. So a lot of good rodent models to study type 2, and I think this is one that I really like to show, because this slide from Roger Unger is one of the more famous biologists. He's probably in his 90s now and still active. This slide's probably 30 years old, and it still illustrates the progression that you get in a mouse, a rat, or a human as you go from lean to obese, from obese to insulin resistant, from insulin resistant to type 2 diabetic. And so what you see here is a rat when it's relatively young and still lean. Insulin is stained for in yellow here, and the acinar tissue or the other pancreatic tissue is in red. Triglyceride accumulation measured biochemically is the number you see above the island. All right, when they become insulin resistant, you can see at first that pancreatic beta cell population expands to compensate for that increased metabolic demand. They're storing a lot more lipid in lean tissue, right? You want that lipid in adipose tissue, now it's in lean tissue. As they continue to overeat and really just get ridiculously obese, at this point they really just sit in their cage right under the food source and just chomp away and don't really move. Right? So true couch potato model. And what you can see at this point, while they're starting to store really a large amount of lipid in islet, the islets take on a very different morphology and ultrastructure. If you pull them out, they're essentially non-functional. And so this slide was from Dr. Unger 30 years ago. Mike Krostad and I collected some of these islets because at first we saw this and thought, well, it's just one islet. You know, was Dr. Unger really being representative of how large these islets were? And it turns out he was, because if you pull the islets out, and this is just the islets floating in culture, so you can study insulin secretion, inflammation, various signaling mechanisms. You see the islet heterogeneity. There are some large ones, there are some small ones, there are some medium-sized ones. That's true in mouse, rat, and human. That's normal islet heterogeneity. If you look at the fat rat, they're almost all large. Right? They still have the heterogeneity. Many, many, many more large islets. And this is exactly what you see as characteristic of insulin resistance. That islet population initially has to expand. When we picked this, these are litter mates, and they're non-diabetic. Right? They're insulin resistant, but they're not diabetic yet. So this expansion occurs as a result of insulin resistance, but then leads them down a path through inflammation to non-function. And so I'll show you what that looks like. Now if you compare, what we're going to do is compare inflammatory markers in this population with inflammatory markers in this population. All right, so this slide's a little bit out of date. There's many more ends here, but the trends are exactly the same. The lean mice cluster together, the fat mice cluster together, and in this case what we've done is just correlated body weight to cycle thresholds. And so cycle thresholds measure gene expression, lower numbers indicate higher expression. So this CD11B is a marker of immune cells. So what we're seeing is, as they get heavier, immune cell population in pancreatic islet increases. Same thing with this marker. So this is two different markers for immune cells. Same concept. As they get heavier, immune cell mass increases within that pancreatic tissue. The reason that interested us is that's exactly what you see in a human situation. You take human tissue, so acinar tissue out here, islets stained for by insulin in red. The arrow indicates immune cells, right? So lean individual, plenty of insulin, small islet. maybe 1 or 2 immune cells. When you start getting tissue from type 2 diabetic islets, you can see they're already larger, there's already less insulin, and there's already more immune cells. So what you see in the obese condition in the rat is mirrored what you see in the obese and type 2 diabetic condition in the people. And so what happens if you look at the function, if you look at insulin output in response to a glucose challenge, this is data in the human population, so here's a number of human subjects, work from Steve Kahn's group. Normal glucose tolerance, you give them a bolus of glucose and measure insulin 30 minutes later, huge response. Exactly what you would expect from a normal set functioning pancreatic islets in a living human. If they've been clustered clinically already into either impaired fasting glucose or impaired glucose tolerance, so this group has been clustered together, there's already a 40% decrease in the ability of that pancreatic islet to secrete insulin in response to a glucose challenge. In a population of human subjects that are already diagnosed with diabetes, 70% reduction. So by the time you're diabetic, there's a 70% decrease in your ability to stimulate an islet with glucose and have it secrete insulin. And so we think a key component of pancreatic beta cell dysfunction is infiltration of immune cells into pancreatic tissue and impaired insulin secretion. And so you can think about how that looked in type 1 diabetes, which is a very different situation, lean, autoimmune onset, versus obese, non-autoimmune onset, both inflammation-associated pancreatic dysfunction. So this handy graph also from Steve Kahn's group really just shows you that if you want to get from normal to impaired glucose tolerance to type 2 diabetic, which I'm assuming we all want to go the opposite direction, you have to limit insulin secretion. So in this case, as someone's insulin sensitivity decreases, as long as you can secrete plenty of insulin, you can stay out of the diabetic range. In other words, you can make 3 or 4 times more insulin than a lean person, and you won't be diabetic. But the minute you can't do that is when you progress to type 2 diabetes. And so a decrease to impaired glucose tolerance, and then a full decrease of 70–80% to type 2 diabetes. And so that relationship of insulin resistance and insulin secretion, I think, is mirrored really well in this graph, and essentially shows you that obese individuals that are insulin resistant and walking around but are not diabetic have to produce 3 to 4 times as much insulin to maintain the same blood glucose level, right? So that's essentially one of the things that drives that huge increase in pancreatic islet size and hormone production, right? So the real question for obese insulin resistant individuals is, how long can you run that engine in the red before it breaks down? And essentially, that is what will dictate the progression to type 2 diabetes. All right, so this slide, which is from Mark Donat's group in Switzerland, essentially points out a little bit more detail. In other words, we know that immune cells are involved in creating insulin resistance in fat and liver. We more recently know that immune cell, and particularly immune cell-derived cytokines, impair the ability of pancreatic beta cells to secrete insulin. And so if you target some of these responses, and the Donat's lab did a really nice clinical trial they published in the New England Journal of Medicine, where if you give an antibody trap, which is essentially just giving a protein that binds up all of the IL-1 beta that immune cells would be releasing, what effect does that have on a type 2 diabetic population? It essentially starts to restore their insulin secretion and improves their glucose tolerance. Right? Essentially showing in a large human subject base that at least one cytokine is directly involved in contributing to beta cell dysfunction. We took that a little bit further and just used a reductionist-based approach and asked the question, if that's true, if insulin's a small secreted protein, which it is, and chemokines, which recruit immune cells into pancreatic tissue, are small secreted proteins, which they are they reciprocally regulated? And the answer was yes. So if you take a normal cell that's not exposed to any of the IL-1 beta, it'll secrete insulin very well in response to glucose, 10 to 12-fold. And production of chemokines, very low. The minute you expose the beta cells to interleukin-1, within 6 hours, they're 40% decrease in their ability to secrete insulin, and have now driven a large increase in chemokine production. So as soon as the beta cell sees this cytokine, it stops secreting insulin and starts secreting proteins that allow it to communicate with the immune cells. So this goes back to the human condition where we think the extra immune cells that you see in pancreatic tissue are certainly secreting these types of molecules, promoting, so in this case they're measuring CD68 positive cells, which is exactly what this particular protein recruits. So mouse, rat, human appear to use the chemokine system, which we saw in type 1, as an example, to recruit immune cells into pancreatic islets. All right, so progression to type 2 diabetes. If you're just now waking up, this is what you need to think about. Overeat, don't move around ever. That'll start by making beta cells expand, you'll be insulin resistant. Circulating insulin will be 3 to 4 times higher, but you won't be diabetic. However, that increased insulin production and circulation will promote increased lipid production in adipose tissue. It'll also promote lipid storage in the liver. It'll also promote lipid storage in other tissues. Adiposity will increase, and at first that will be good because you want fat to go in fat cells. You don't want it going in lean tissues. But the minute you start having to put fat in non-adipocyte, which is what's referred to as ectopic lipid deposition, really ramps up insulin resistance. But you can't get to type 2 diabetes without a loss in pancreatic islet mass function. That's really the negative outcome that transitions insulin resistance to full type 2 diabetes. All right, this shows you a little bit of what happens in the islet in human subjects if you have type 2 diabetes or you don't. Give glucose, and what you can see here after a meal, in this case they're measuring glucose after a meal, rises, but it rises in the normal individual. But in the normal individual, you see it peaks and comes back down. You have insulin production. No problem. You push it into tissues, stop liver production. In the type 2 individual, it kind of just stays up. And the difference is, during that meal, the normal individual spikes their insulin production, and as glucose comes down, it drops. But the type 2 individual doesn't have that ability anymore. And you can see the other major problem is the glucagon response. Normally, you need insulin to go up and glucagon to go down. In the type 2 diabetic islet, if anything, a meal triggers glucagon release, which it should not do. It should shut off glucagon release. Right? So you don't have enough of the hormone you need, and you get overproduction of the hormone you don't need. If anybody read the review I sent out, you'll see that if you want to cure type 2 diabetes, if you want all of us to be out of the job, have everybody eat right and exercise because there wouldn't be any obesity. And you can see that experiment's been done. One of the biggest clinical trials in human history done accidentally. Put people on rations during World War II. They don't have enough to eat. There's no obesity. Guess what? Type 2 diabetes, non-existent. Incidence of type 1 diabetes, totally unaffected by food availability. The minute rations are restricted, war's over. People can eat as much as they want. Obesity rises, type 2 diabetes rises. All right, so the final summary here. You've got a normal population of beta cells. You become insulin resistant. We know that there's an inflammation component, but not all the details and all the signaling mechanisms. Those are still being actively pursued in many labs around the world, including at Pennington. Initially, there's an increase in pancreatic beta cell population, coupled to an increase in insulin output. This will keep you from becoming diabetic. However, if this continues unabated, what you get is increased immune cell infiltration, provides an inflammatory response that ultimately produces a demise in insulin secretion, reduction in that pancreatic beta cell population, and that's how you get type 2 diabetes. From a type 1 perspective, almost all of the individuals are lean, and they have autoimmune mechanisms that target their beta cells that produce type 1 diabetes. And so what my lab essentially does is works on the inflammatory mechanisms here and here to ask what are the similarities and what are the differences, because the end goal is ultimately the same. All right, so I'll stop here, and if there are any questions, be happy to take those. Yep.
What about lean type 2 diabetics that are not obese but develop diabetes? And that would kind of go, say it's in this order, pathology goes in a certain direction, where,
Where they fit?
yeah, where do they fit?
Almost all of those fit on a spectrum of inherited mutations that affect the ability to secrete insulin, right? Because ultimately what's going to control blood glucose levels are the interplay between island hormones. You want insulin going up after a meal, you want glucagon going down. If you don't get that shift, you'll become diabetic. And that's true if you're lean, or if that's true if you're obese, right? But there's at least 40 different mutations with different degrees of severity that affect the beta cell's ability to secrete insulin in response to glucose. It could be a membrane channel, it could be in a metabolic enzyme. The lean individuals almost always fall into those forms.
Is there a selective reduction in first phase or second phase insulin secretion when it comes to hyperinsulinemia over the course of, developing adiposity and all that? Is there,
So
or
first
are they
phase
kind of
is
both
almost,
equally effective?
first phase is almost always wiped out,
Yeah.
right? And if you look at an insulin secretion curve, and it's going like this with no glucose, and then you hit it, there's a huge first phase,
Mhm.
right? And then the action potential is over, and as the cell hyperpolarizes and repolarizes, then it's able to be stimulated again, and that second phase is kind of a continuous oscillatory pattern. If you look at lean individual and type 2 individuals, both phases are blunted in the diabetic or the obese or the insulin resistant, but first phase is even the most blunted. So it's that initial burst that they don't have.
In type 1 diabetes, if you have a patient that were to for some reason become immunocompromised, maybe a transplant or something, and they're on immunosuppressants, do you see recovery of the beta cells over time?
That's a really good question. So, yes, is the short answer. So that experiment got done with cyclosporin, which is a really powerful immunosuppressive drug, and was used in many graft rejections. It's obviously been phased out because it has nasty side effects. But with new onset type 1 diabetics, as they were essentially figuring out it was a relapse-remitting disease, if you gave them cyclosporin, you essentially restored beta cell function, right? Which let people know there was absolutely an immune component involved. And that's really what triggered the major studies of, hey, what's this beta cell immune cell interaction about, and how does it work? It was those early studies all on immunosuppressive drugs that really brought that out.
Do you see that with patients that are 30 years out with type 1 diabetes as well as that are only newly onset?
The patients that have had long histories of disease, the ones that die eventually donate their tissues for research. There's still a very, very small population of insulin positive cells, so they never get 100% elimination. But the immunosuppressive drugs aren't thought to work in that circumstance because all of the cells have largely been eliminated, so there's nothing to recover from, right? So the idea would be if you still had 50% and you stop the response, could that 50% grow back to 80%?
Gotcha.
Yeah, nobody knows, but people think maybe. Well, if you get to 80% or 90%, how are you going to get that to grow back? And I think that's really one of the challenges. I mean, there's a number of NIH, JDRF, ADA grants funded to try and answer some of these questions in mouse rat, and clinical trials obviously ongoing for different mechanisms related to those concepts.
If you have a person that's hypoglycemic, is that also considered to be diabetic?
Hypo?
What do you have? Yeah, low blood sugar.
Diabetes is usually the opposite, right?
Mhm.
It's hyper. So hypoglycemia is a sort of a different condition, which can be from a variety of different mechanisms. The interesting thing are some of the cases of hypoglycemia are triggered by beta cell mutations that do the opposite, which secrete too much insulin. Right? So if you start pumping out too much insulin and you're a really insulin sensitive individual, you don't really allow any production from liver, which you need to sustain a normal level, and you're driving all of it out of blood into tissues. And so that blood amount drops way down. And so a lot of those are treated with drugs that actually block insulin secretion and allow that glucose level to rise back up.
In the case of patients that have bariatric surgery, I know that in most cases, if they do have type 2 diabetes, that can be reversed with the bariatric surgery. So I guess in that specific population, is ectopic lipid deposition also reversed?
That's a really good question. So, there's some really good, so the bariatric surgery situation, regardless of whether you're talking about the gastric banding or the Roux-en-Y gastric bypass procedure, you're limiting the amount of intake someone can have, right? If you take a human population and give them a very low calorie diet, 600 kcal per day, you can essentially mimic all the outcomes of bariatric surgery. So what you're essentially doing is, A, taking stress off of beta cells because they're not having to respond to nutrient stimuli over and over and over for a period of years, right? So part of the reversal is lowering constant stimulation on the insulin secreting cell and allowing it to essentially recover, for lack of a better phrase. And if you are no longer over consuming calories, you're forcing the normal homeostatic mechanism to then start burning calorie to compensate for that difference, right? So to get at the question of ectopic lipid deposition, it absolutely clears fat out of liver, out of muscle, out of places you don't want it, and restores insulin sensitivity, which is exactly what you do want. And so it shows really the power of decreasing caloric intake and really allowing the body to go through the appropriate periods of feeding and fasting to allow the hormonal mechanisms to operate appropriately.
Also works with less strict diets. The calories not really repetitive here, so 25% calorie restriction. Also non-obese people.
Sorry, at the beginning you said that diabetes is not highly related to genetic.
It's highly related. I said it wasn't
Okay.
100%.
Okay, so how much is the risk to development diabetes when you have some health history, like family history about diabetes?
You're saying, does family history of diabetes increase your risk?
Yes.
Yeah, absolutely, right? I mean, there's definitely a genetic predisposition. In other words, you can inherit genes that either predispose you to insulin resistance, or predispose you to obesity, or predispose you to more poor beta cell function than a normal individual. That doesn't mean you're definitely going to have diabetes. You really have to push the system with some of these other mechanisms. In other words, chronic overnutrition and lack of exercise, for a better term, to start pushing it in the direction. You need the genetic risk on top of the environmental factors, right? So in other words, genetics don't explain everything unless you have a monogenic form, but most people don't have that.
And the other question, sorry, is about, you were talking about the administration of insulin when they have type 1 diabetes. So how much is that? It depends on each person, how they...
It absolutely depends on the person, right? So, and it depends on not just the person and what their body mass is. It depends on their body mass, what their diet is, so how much they exercise. In other words, multiple factors go into deciding what's the optimal dose for keeping you in a normal blood glucose window, but not going into hypoglycemia. And so when we were at Duke studying some of these individuals, if they wanted to go exercise, they would let their blood glucose get up to about 350, and then they would work out for an hour and they would be back to 120. But if they would have given a dose of insulin, they would have overshot it, right? So physical activity decreases the amount of insulin you need, right? Sedentary behavior will increase the amount of insulin. Certain other drugs will increase the amount of insulin you need. So there's a lot of factors that go into how many carbs you eat, for example, because all of that sugar has to be handled in some capacity. Well, if you don't have functional beta cells, you don't have minute-to-minute control of glucose. So you have to time the dose of insulin with a meal so that everything's regulated properly. So body mass, dietary components, and how much exercise, I think, are really three of the major factors that determine dose.
Thank you.
Yep.
So how do you view gestational diabetes? Is the predisposition there in the pregnancy is the trigger of the insult then?
Right, so it's a totally different category. And pregnancy as a state involves insulin resistance normally, right? Because part of the insulin resistance of pregnancy is you need the mother to be slightly insulin resistant so that the fetus can absorb more of the nutrients, right? Because essentially you have competition for nutrients between fetus and mother. With mother becoming slightly insulin resistant, it gives the fetus an advantage, which is good. It's when that slides too much in one direction that the blood glucose starts to rise. In other words, if you're predisposed to insulin resistance, you might have a higher risk factor for gestational diabetes. And then if you have gestational diabetes, and then after birth you're normal, your risk for type 2 diabetes later is higher, right? And so there's some interrelationships there, even though they are different, because of the pregnancy hormone differences that aren't present in the non-pregnant state. You know, particularly glucocorticoids, growth hormone, you know, progesterone. There's a lot of things that aren't spiked or aren't elevated when you're not pregnant that are when you are pregnant.
Another question. You mentioned that there is T-cell infiltration to the islets in type 1 diabetes. Do those T-cells have roughly the same T-cell receptors? Like, are they antigen specific, or is it just general recruitment of T-cells?
They're antigen specific. So they're still primed like they would be for a normal infection response, except for in the autoimmune situation, they're primed against the self-tissue, right? Obviously, proinsulin is one of the major priming factors. GAD65 is one, zinc transporter 8 is one. So there's different T-cell population that, for whatever reason, are now primed against the self-tissue. And we think coupling beta cell production of chemokines with that priming by autoantigens, when they show up, they're ready to do business.
Yeah.
And, you know, they obviously start to eliminate the cells you want to keep. But it is. Any other questions, comments? If not,
That was excellent.
great.
Thank you.
All right, thanks
for that.
everybody.
Yeah.